friends, welcome back to another video tutorial from Shomu's Biology. And in this video tutorial, I am going to talk about transposable elements. Now, I received all these requests from different students and subscribers about transposable elements. That is so much confusing because the idea is easy, but there are so many names and so many types of classifications. So how to exactly figure out what kind of transposons we're looking at? We have TN elements, we have TY elements, P elements, ACDCS elements. So what are these things? So to answer all these questions in one frame, that's why I'm making this video. I already had video on transposons earlier, but this is to summarize everything regarding transposons. So in this video, I want to talk about what are transposons, especially the classification of transposons and what kind of transposable elements that we have. Actually, it's better to say them transposable elements, because transposable element means it's a broad scale. If we divide them down, then one part is a transposon. And we also talk about the mechanism with which transposable elements work and their functions and their requirement in the modern research. So let's talk about them. First of all, transposable elements have the unique capability of duplicating themselves or transferring themselves, duplicating or without duplicating, from one region of the DNA to the other region of the DNA. So this is kind of a definition. So the idea is they will transfer themselves from one part of the gene, one part of the DNA to another part, one part of the chromosome to the other part. Now it can be transfer between the same DNA, same chromosome, or can be between the different chromosomes. That's why they are also known as jumping genes, because they can take a jump from one part of the gene or DNA to the next part of the DNA. So let's look at the classification of these transposable elements to get an idea about that. So the most important property for a transposons is the ability to be transferred, okay, intergenomically. Now this transfer can be utilizing duplication or non-duplication. Now if I break these transposable elements down, we can have mainly three different groups out there. So let's draw it. Three different groups we have. One type of group we have viral transposon groups. Second type we have is non-viral transposon group, and the third one we have bacterial. So simply if we divide bacterial group, this is mostly found in bacteria, but it does not mean it's only found in bacteria. It's also found in eukaryotes also, but we'll see that. But the grouping is based on the type of element and their structures and mechanism of transposition. That's why we group them. Okay. Now there are different way of grouping and classifying, but this is one of the unified way of classifying. That's why I'm interested in that. So in bacterial group, the elements that we know is known as IS elements. These IS elements are part of bacterial group. This is found in bacteria. Let me write where we find these IS elements. We find in bacteria, okay, different bacteria will find that. We'll also find that in Drosophila. We find that in corn or maize, whatever you say. We find these IS elements in all. So you see, we find in plants, in eukaryotic animal, as well as in bacteria. But the type is a bacterial transposons known as IS elements. The second type, non-viral, and third one, viral. So the viral type of transposons are known as LTR transposons, or LTR elements you can say, because they contains LTR at the end. This is a terminal repeat at the end of the transposons, because you know transposons, what are these transposable elements? They are a fragment of DNA itself, capable of transferring from one part of the DNA to the next, one part of the chromosome to the other part of the chromosome, between the same chromosome or different chromosome, right? So in those DNA, they have a flanking repeats, and that's the property of transposable elements. Every transposable elements contains this kind of repeats, but the repeat is varied. For example, for IS elements, there is a specific repeat. For LTR, this is the one type of repeat. For non-viral, there is a different type of repeats. So these are also known as LTR elements, and these LTR elements are found in they're mostly known as retroviral transposons or retrovirus elements. But not only they found in viruses, but also they are found in Drosophila, and the example is copia elements in Drosophila, and it's also found in yeast. In yeast, TY elements are known as this viral transposable element family. While if you look at the bacterial one, it's found in bacteria, it's only known as IS elements. It's also found in Drosophila, known as P elements, and it's found in maize, known as ACDS elements. So the name of the elements are listed along with the name of the organism where we find them. And the third and last one is the non-viral type. It is sharing a very similar mechanism with that of a viral transposition, but these things are mostly found in eukaryotes and higher eukaryotes. In humans, we'll find a lot of this non-viral type of LTR, non-LTR element. We call them non-LTR elements. This non-LTR type, instead of LTR, they contain UTR. Also divided into two different parts, so let us divide it into two parts. This is mostly found in higher eukaryotic organisms like human being, for example, and the example for that is lines and signs. These are the two different types that we have. Signs are also known as ALU. This is known as ALU, so they are known as ALU elements, very common in human genome. Now it's time to understand about the mechanism, because one thing, if you remember, you'll see that those bacterial transposons, non-viral and viral transposons, but actually they are not quite only for bacteria and viruses. It's kind of broadly separated around all these different types of individuals. So the actual question is why and with which type of property we are actually separating the different type of transposons. And the answer for that is the mechanism of transposition, how the transposons work. Now the simplest idea about transposon is the job of the transposon is to cut itself out and put it to another region of the genome. Now they can cut out and paste, or they can copy themselves and then get spaced. So either this is a replicative transposition or non-replicative transposition. So based on this way they transpose is replicative and non-replicative. Okay, so normally replicative transposition can work like if there is a DNA fragment. If I say this is the fragment of the DNA, and let's say this is the part known as the transposons, this is the region, that region, and let's say the target DNA, this is the target DNA, this is the region of the target. So what it does actually, this part makes itself a copy and then gets embedded inside there. That is a replicative transposon. So you can replicate the fragment of the DNA and attach it to the other target DNA. While a non-replicative transposon simply can cut this fragment of the DNA and put it there. So for a non-replicative transposons, the donor will get shorter because the fragment gets cut out, and the recipient will be lengthier. While for a replicative transposons, donor remains same as it is, of same length, while the target receives it, so target gets lengthier or taller or longer. That's true for all. Now if you look at this simple mechanism of cutting a DNA and then pasting it, or copying a DNA and pasting it, it requires two different tasks. One is the endonuclease activity, and second job is ligase activity. These are the two enzymatic activities required, endonuclease and ligase. For a cut paste transposons, endonuclease will cleave the target DNA, ligase will seal it. Now in this case, both these activities are mostly played by a same part of the enzyme known as transposase, and the gene codes for those proteins known as transposase gene. The simplest form of transposons and transposable elements, if you look at here in this picture, is this IS elements. And this IS elements contains nothing but one gene only as a transposons, known as transposase. So the mechanism we saw just now, it's the mechanism of IS elements containing only transposons, no other genes inside. Simply cut it and paste it. That's the job. So no other structural genes or antibiotic resistance, nothing is there. Simple. That is the IS elements. Now the job of IS elements is not very properly understood because it's not giving any benefit to the bacteria. But if this IS element, between two such IS element, it contains fragment of gene that carries antibiotic resistance feature, or that helps to produce antibiotic resistance proteins. That case, bacteria will get the benefit. So we will also find those things, that such type of transposable elements also there. So this type of elements contains two, it contains antibiotic resistance gene, for example a tetracycline resistance gene. That kind of modified IS elements are very important in perspective of bacteria, because once they receive this antibiotic resistance gene, using these transposable elements, it can transfer this antibiotic resistance gene from one part of the cell to the next, and also can transfer it to the other individuals there. That can help them to make a huge cassette of antibiotic resistance, and we call them integrons. These integrons are a cassette of this such antibiotic resistance received by and with the help of this jumping genes or transposable elements in bacteria, and as a result those bacteria gathered a lot of such type of more antibiotic resistance genes, added one after another, make a chain of antibiotic resistance gene island, is mostly called superbugs. Example, MRSA, methicillin resistant staphylococcus aureus. Those kind of bacteria, they contains a huge array of antibiotic resistance. They get that with the help of this kind of transposable elements. Now this type of transposable elements which contains extra gene apart from transposase gene are known as composite transposons, because these transposons flanking with IS elements and also contain extra gene, composite transposons. While there is another variety of transposon known as non-composite transposons, who also carry extra gene, but the terminal part is not IS. The terminal part is inverted terminal repeat, or ITR. Difference between composite transposons and non-composite transposons. Another thing, for non-composite transposons, normally it's bigger, lengthier, while composite transposons are a little shorter. Normally non-composite transposons can have like 5,000 base pairs of length of materials inside in between. So these are the two differences in that perspective, composite and non-composite, but very similar with the part of the IS elements that we saw earlier. Example of this kind of IS elements, as I told you, in bacteria it's normally known as IS elements or composite transposons. Different composite and non-composite transposons, for example in Drosophila we have these P elements, that is a type of IS elements. In case of maize we have ACDS element. Now these are the elements which are also this type of composite and non-composite form. Now the question is, normally in case of ACDS element this is a non-composite form of transposons. Now the question is, there is another difference you probably heard. One is the self-dependent transposon, and the one is the self-independent. Self-dependent transposons are autonomous transposons and non-autonomous transposons. What are they? Again, same range. ACDS element, if you take, among these ACDS element, both of them have the very similar feature with the non-composite transposons, but this AC elements is known as autonomous elements, while DS element is non-autonomous elements. Why? Because autonomous elements are known as those type of DNA or transposable elements which can translocate themselves easily without the help of any other gene. So simply cut it out and paste it someplace else without the importance and help of any other gene. But the non-autonomous one, for example DS elements, DS elements is a non-autonomous transposon. So DS region of the DNA to the other region, it requires the help of AC element. That's why it's a non-autonomous transposons. So if you wonder about what is composite, non-composite, autonomous, non-autonomous transposons, this is the thing, and all of this thing placed in the IES elements in the bacterial type of transposons. While the next type is this LTR elements or viral transposons. These LTR elements, the difference with IS element is simply they also contains different genes in the middle, but here the terminals LTR. These are different types of nucleotide repeats, and this repetitive DNA is found in human genome is so much amount. For example, in human you will also find this LTR type. The example is THE1. THE1 or D1 is one type of LTR elements that you find in human, and they have a functionality of a retroviral transposon. You know, retroviral transposons, why? Because these are not generally cut paste, these are copy paste transposons. So what they do normally in retrotransposons, there's a DNA that needs to be transferred. So what they make, instead of copying the DNA using a replication, they produce an RNA from the DNA with transcription. Once they make this RNA, then they use this RNA to make a DNA, and we can prepare DNA from RNA using what we can use in this case, reverse transcriptase enzyme. And once we use reverse transcriptase enzyme here to replicate RNA into DNA, that is known as a retrotransposons. And this thing is mostly found in retroviruses, and it's also found in eukaryotes, as I told you. For example, in human THE1 element, in Drosophila Copia elements, in yeast TY elements. All of them, they carry these extra genes in the middle, but they also have these LTRs in both the end that will help them, and also they produce reverse transcriptase enzyme that helps them to replicate, make a DNA copy, complementary DNA copy, then put it there in the target DNA. While the third type, or non-viral or non-LTR type, instead of LTR they contains UTR, as I told you. And this non-LTR type retrotransposons are very important features for eukaryotes, higher eukaryotic organisms. They are not found in bacteria and very less in plants also. They are mostly found in high eukaryotic animals like mammals, like humans. In humans you'll find a lot of these lines and signs, long interspersed nucleotide elements and short interspersed nucleotide elements. So signs are shorter, lines are lengthier, but these are repetitive sequences in stretch. This interspersed repetitive sequence that we know, which act as a marker also, we can use them as a marker to find out different properties and differences between individuals in a population. I also had a different video about these molecular markers, IISR, SSR and all these things. You can watch the video. But here you know, the signs, they are also known as ALU elements. They are present almost 10% of all the transposons that we know in human. Lines represent 20% of all the transposable elements and repetitive sequence that we know in humans. But all these cases, you know, they contains a specific type of structure which is different from both elements and LTR elements, because they can either have UTR in both the ends and all the reading frames in the middle, or they also have different ORF, reading frame 1, reading frame 2, and they also have poly-A tail. All these lines and signs, they have this poly-A tails at the end. Now they have either UTR present or not, but they have a poly-A tail at the end. So this is the typical characteristics of this eukaryotic processing of the RNA. And this type of structures, from this ORF, they can also produce different components of protein. So they make different proteins that can help them to cut from there one place and place it someplace else. But the importance of these lines and signs in eukaryotes like humans are very much very important, because these fragments can jump from one place to the other, and that gives a variation. So transposons in perspective of the organism, in case of bacteria, provides antibiotic resistance. In perspective of eukaryotes like Drosophila, yeast or human being, it helps us to give rise different variations, modifications even between two individual human beings. Though according to the theory everything should be same 100%, but actually there are slight modifications due to these changes, due to these modifications of repetitive elements and jumping genes like lines and signs. So you see, most of the part of our genome, of our eukaryotic genome, are made up with these lines and signs and all these elements earlier known as junk DNA elements, which are not the part of exons but the part of introns, normally cut out, but sometimes they also plays a vital role throughout the evolution. If you check them earlier, some part of the time they definitely had some role, but right now they mute those roles, so only few part is involved making proteins, but these other parts are also important to make who we are today. So that in a sense is about transposons, and I hope you understand transposons in a whole. Remember, learning everything as an overview is the most important part, even going to the details. So I hope you know and get the idea about the transposable elements. If you like this video, please hit the like button, share this video with your friends, and subscribe to the channel to get more and more videos like that. Thank you.